All right, ladies and gentlemen, welcome back. So today we are going to pick up to talk about the internal structures of your cells. And today let's just talk about what gives the cell its structure in the first place. So when we're looking at cells, they're not just sort of these gelatinous masses um, floating around, but they do have internal networks, internal structure to allow them to anchor things in place, to move themselves around, to move things internal to their cell around. And so, what we're talking about here is your cytoskeleton. It literally translates into cellular skeleton. So, just like your bones and attach to muscle and allow you to move and give you support and give you structure, that's exactly what these fibers are doing for the cell. So, when we look at these, they're protein fibers, and they're put together in a variety of arrangements that really kind of stand out and give us three major types of fibers. Your microfilaments, you may also come across these as actin fibers. If you've ever heard that term actin before, it's probably in connection with muscle. You've got a lot of that same fibrous protein going on in your muscle cells. Microtubules, and then intermediate filaments or intermediate fibers. So let's take a look at each of these. So when we talk about microfilaments or these actin filaments, they are, in terms of scale, the smallest of your cytoskeletal fibers. When we actually look at these under a microscope, they sort of look like two pieces of yarn wrapped around each other. Um, and their biggest thing is to really do a lot in your cell membrane. Remember, that membrane is going to be that outermost covering of your cell. And so it's going to anchor things like membrane proteins. All right, when we get to talking about cell transport, we'll talk about those membrane proteins, among others, that sit in that membrane and have a whole handful of jobs to do. We want to keep them in place if we want them to be in certain areas in order to do that job, so those microfilaments can help us keep them there. And we can also expand and contract these filaments to pull the plasma membrane around. Um, so, in the situation of cells dividing, we'll talk about how do we separate the cytoplasm to make two separate cells. Microfilaments are going to play a role there. In cells that sort of do amoeboid like movement, meaning they sort of like shove part of their cell membrane out in order to move around, these filaments are also there for that sort of purpose as well. Your microtubules are on the opposite end. These guys are the biggest. They're long, hollow tubes, kind of remind me almost of straws in terms of the way that they're built. So they're hollow in the center, and then their proteins go around the outside. And these guys also help to enable the cell to change shape and to move. And that is both on the external side, so the sort of cell membrane component. They are also part of your cilia and your flagellal structures, which we'll talk about in a moment. These also run all internal to your cell as well and sort of act like a highway system or tracks for those organelles to move around. In fact, we'll attach organelles to a protein called a motor protein, and it kind of almost looks like a little walking stick. And so the organelle sits on the back of that protein, and then it's got almost like two hands that sit on these microtubule tracks and grab and release and pull themselves along. It's kind of cool to look at. Um, we also see these guys dragging other molecules around, so on and so forth. So these guys are really big for movement, both the cell as a whole, but even more importantly, for all cells that have a cytoskeletal structure, these guys are huge in moving things internally around as well. And then you've got your intermediate filaments, which literally sit in between the two. So microtubules are the biggest, microfilaments are the smallest, and then the intermediate filaments, size-wise, sit in between. These guys also kind of look like straws in terms of their structure, but they would be smaller straws. So if you've ever seen, say, maybe one of those big honkin' straws from like McDonald's or something, these might be more on the order of something like the size of a coffee stirrer. where they're much smaller, but they're not. They still have a hole in the center. Your your microfilaments do not have that hole in the center. These guys are mostly for reinforcing cellular structure. They're also going to anchor certain organelles in place so they're not kind of sloshing around. And then they also do help to organize complex metabolic activities, aka chemical reactions, by ensuring enzymes and certain chemicals are close enough to each other inside the cell to ensure that they are going to be able to interact with each other and go through those chemical reactions as efficiently as possible. All right, while we're on the topic of your cytoskeleton, let's talk flagella and cilia. Some cells have these, some cells don't. But when they do have them, they are made of microtubules, so those biggest filaments of your cytoskeleton. And it's just a matter of how long are they and in what kind of arrangement. When we look at your flagella, right? These are going to be these long, sort of whip-like structures. Um, they are made of clusters of microtubules, and they are there obviously for cells to be able to swim around. Um, some cells can have a single flagella, some can have multiple flagella. Some of them will have them coming out of one location, some of them will be out of multiple locations. So it really just kind of depends on the type of cell you're dealing with, right? Remember, we also see flagella in prokaryotes, although we actually can look at them. They do have some structural differences in the way that that protein gets built, but they use them for the same purpose. Only cell in a human body that has a flagella? Sperm cells. All right, so they'll use their flagella in order to swim to find that egg and actually complete fertilization and make an offspring. Um, when we look at cilia, these guys are much shorter. They're still microtubules, but they're much shorter than those flagella, and they're typically all along the cell. You'll see them kind of look like this. In a cell that is capable of movement, like our little paramecia here, they'll use those sort of whip them around like motors, rotors, I should say, on a motor, and be able to make themselves move in a particular direction. Um, there are cells, though, that are stationary, meaning they personally don't move, but they still have cilia. And in that case, they will use those cilia to beat fluid over their cell surface for one purpose or another. In our body, the cells that line your windpipe going into your lungs actually have cilia. Those cells don't move, but what they will do is move mucus and other dirt particles and materials um, across their cells, not really for any purpose for the cell to gain nutrients or anything like that. But what they call it a ciliary escalator. And what that really means is that they're constantly beating back up so that anything that tries to get into your lungs will hopefully get swept back up your windpipe and be able to go down into your esophagus and into your stomach, so that you're not getting things like bacteria or dirt particles or things like that deep into your lung tissue where you really don't have a good way to get rid of it. Um, so it's a pretty cool system to try and keep your lung tissue healthy. Unfortunately, people who are chronic smokers, for example, who or have some other potential lung issues, one of the things that can happen is that all of that gunk and things like that can basically arrest or freeze up these cilia and make them not capable of beating anymore. They kind of get gooed up. And when that happens, that ciliary escalator breaks down, and now you're getting dirt particles and um, bacteria and things that end up sitting in your lungs. And a lot of people who have those chronic problems like that um, end up with a lot of bronchitis, and sometimes that can turn into pneumonia, and you get fluid buildup in your lungs. So we definitely want that ciliary escalator to be doing its job as best as it can.